pleasure to welcome Jörg Stelling. So he received his master's degree in biotechnology from the Technical University of Braunschweig in Germany in 1996, and then the PhD degree in control and dynamic systems from the University of Stuttgart in Germany also in 2004. And from 2005 to 2008, he was an assistant professor with the computer science department at ETH in Zürich, in Switzerland, and he has been now a tenured professor with the Department of Bioscience, Biosystem Science and Engineering at the ETH since 2008. And his research interests include the analysis and synthesis of biological networks using and further developing methods from system theory, computer science, and experimental biology. So whenever you want to start, Jörg, the floor is yours.
Thanks a lot, Marie, for the introduction, to all organizers for the invitation. So the topic of this talk is about microbial communities and how to use mathematical models for analyzing them. But I want to start, since this is the first talk, with a little bit of background, a little bit of motivation. So one aspect where we have a lot of unknowns in microbial communities is essentially the gut microbiome. So we're involved in a study on IBD, so inflammatory bowel disease cohorts analysis. And usually what is done in this field is you have a patient, you take a biopsy, submit this to 16S sequencing, and map this to taxonomy. So what we get in the end, or that's the stopping point for most studies, we have the microbial abundances, and then we need to, for example, make sense of this in terms of what is characteristic for one disease or the other. So here we were interested in discriminating between ulcerative colitis, UC, or Crohn's disease, CD. And the question we asked is, there's simply a better discrimination when we look at the taxa, or when we use models and look at the abundances, so the capabilities in terms of reactions that the entire microbiome can have. So essentially what we did was just take genome-scale metabolic models, counted the number of reactions, and multiplied this by the microbial abundances. So very simple, so not really mathematical modeling yet, but we'll come to that later. So the interesting finding from the study was here on the top. So these taxonomic units are usually operational taxonomic units, OTUs. What you see here is just discrimination by principal component analysis, looking at the percent variation explained. You see here for principal components 1 and 2, altogether less than 5% is explained. And you can hardly discriminate between UC, which is in red, and CD, which is in blue. If we look at the reactions, as I said, just enumerating, that's the entire trick here, we're close to 55%. And this also reflects itself essentially in this statistical discrimination between UC and CD. And those are the corresponding p-values here and p-values here. So that was one cohort. This could be essentially random, but it was not. So what we found for another cohort as well is essentially the similar picture, less than 10% of variation explained just by taxonomy, more than 50%, in this case 60% explained by capabilities, biochemical capabilities. Because with these more reaction-centric analysis, we can then ask what are the functions that are affected. And that's what we did. So looking at metabolic subsystems, and you see here, again, for cohort 1, that some, like LPS biosynthesis, they are significantly higher, overrepresented in CD. Some others are underrepresented. So the idea here is that we can then use this knowledge in order to try to propose treatment options, seeing which subsystems discriminate between the two. And again, this was not random. Essentially, we see a very similar picture for the other cohort. So this was not really a use of mathematical models. What we try to do more in more detail is the following. So we have this typical model content for constraint-based models. So it's based on the stoichiometry here, N. We have the unknown reaction rates of fluxes in the network. We assume quasi-steady state. And then the models themselves have as content both all the internal metabolic reactions, exchange reactions, and usually also biomass reaction, denoted here by mu. So then what this approach does in general is try to characterize the solution space for this linear underdetermined system. So we come up with a feasible flux space. We can have essentially two main handles to incorporate additional knowledge. So on the one hand, bounds on the fluxes. So for instance, by reaction reversibilities, and then assumptions on objectives. Like in flux balance analysis here, we try to maximize growth subject to all the other constraints, such as the stoichiometry. So these are the two handles that allow us to adapt this framework to specific problems. There's a number of challenges there. I just want to mention two because they're relevant for later. So the first one is, if we want to predict fluxes or responses to fluxes, then this is hard because of kinetics. And this is just one example to illustrate this. We have a little system here. So in influx UA, then just a diverging pathway to B and C. And here you see two simulations. These are just arbitrary parameters. The scenarios, we have full concentration of E1, so the one converting A to B, before time 20, and then we have this concentration. And you see the reaction rates of fluxes here. And the response, if both enzymes are roughly similar in terms of quantities, is what you would expect. Reduce enzyme 1 by half, and then you get more flux through this pathway as compared to this pathway. If enzyme 2 doesn't exist, then the obvious solution is that everything goes through the pathway again. So this is also one of the main difficulties how we want to or aim to integrate genomics data, simply because fluxes are not proportional to enzyme concentrations, and the same essentially holds for metabolomics data. So that's on the data integration side. More on the technical side is, and this is for consortia specifically, that if you are considering a single strain, say this strain here, it can grow. So CI are the corresponding concentrations. Internally, C is the external concentrations. You have fluxes, exchanges with the environment. You also have biomass X here. This is really well captured in the normal traditional FBA framework. But if you have two strain consortia or more, then we do not only have this replication of concentrations in the species, but also essentially all communication goes via the external environment. So this makes the prediction of factors that are interesting, such as cross-feeding between species, really hard. So what I want to show today is essentially expanding on this idea. We have consortia, like this two-strain consortium here. What we can usually do, and this is a very famous approach in the field called OPCOM by the Maranas group, is essentially to assume that there are multiple objectives for consortia. So the mathematical formulation is shown here. What we have here is essentially the assumption that there is a community objective. So this, in this case, it would be the maximization of all biomass produced in the community. We include mass balances. This is shown here. So the fluxes essentially enter here. And then at each level of the optimization problem, we have now the internal or cell-specific or species-specific fluxes. And we assume again that there's a maximization, for example, biomass. But the constraints by the community enter only very indirectly. You see this here. And it's unclear essentially how this relates to the physical reality that in the real consortium, the exchange is via the external environment. So that's what we're trying to address, trying to advance this a little bit more. And essentially there are two proposals. One is to use metabolic pathways for this. So essentially trying to enumerate all possible interactions in a consortium that are consistent with the internal workings and the exchanges. So this is really in the spirit of metabolic pathway analysis, where we try to characterize the entire flux space that is feasible. And from that, we try to make inferences on which interactions are likely to exist or cannot exist in a particular consortium. And then in the second part, I want to show you an even more recent approach here, where we have explicit models for consortia, explicit models for objectives, but without having to have either dynamic FBA type simulations or the need for any kinetic parameters. So first for the pathway analysis part. So the idea here is that we have, for example, a metabolic network. It could also be a metabolic subnetwork. We have defined bounds on fluxes. These are described here. And the idea is now to have a minimal representation of this, the capabilities of this flux space, including both the network structure on the left and the bounds on the right. So there exist many different concepts for this. I will show you the distinctions on the next slide. Essentially, the main ones are so-called elementary flux modes and that don't consider the bounds, or elementary flux vectors, these ones here, that consider the bounds. So the concept, or what we try to do, is come up with something that is minimal, that has no loops, so it's thermodynamically feasible, can span all the bounded flux space, and in addition allows you to pick and choose which reactions you want to consider explicitly and which ones you don't. So that's to make this a little bit more intuitive. So for this little example network here, these would be the traditional elementary flux modes. Here you have these three elementary flux modes, the upper branch, lower branch, and the cycle. Similar for the elementary flux vectors. For the MPs, minimal pathways, you would include the bounds and also remove the loops. So the way how this is done computationally is the following. So we make this definition for arbitrary subnetworks. So they're essentially equivalent to so-called elementary conversion modes, but we can choose any reaction in the network we want to consider. They fulfill the entire constraints on the network. So that's the distinction to EFMs. And they also support minimal subset of the EFVs. So computationally, what we do is essentially iteratively solve linear programs to do this enumeration. What you see here is over time, the algorithm, how it proceeds to, in the red here, generate the fraction of MPs. In this case, it's for a E. coli network, 560 reactions, and you have roughly 27,000 of those. And you see also that we iterate over the subnets considered and also over the sizes of the MPs. So with this, what we essentially can do is to scale this to much larger networks. So on the left, you see the fraction of maximum, again, how the algorithm proceeds. In this case, this was a metabolic network of a human and 6 microbes. So altogether, roughly 30,000 reactions and roughly 3,200 possible exchanges. So we focused just on the exchanges because we were interested in who can produce what for the community or consume what. And then we looked at essential exchanges. So for a given growth condition, we just minimized the number of exchanges, trying to figure out what is essential, what is not. So the outcome for this particular consortium is here on the right. And so on top, we have the human model, then the 6 microbial models. We have in the columns the particular metabolites that can be exchanged. And then red color means that particular substance is taken up by a particular species, blue means produced. And for instance, what you here see is that human mostly consumes. Certain bacteria, such as Lactococcus lactis, produces with very high probability, so meaning with all MPs, isoleucine and ornithine. And you see the interactions within the community. So we try to validate this against experimental data. And so far, this tends to look good. So we have some new predictions that we're actually testing in collaborations. So this was essentially the first part, the first idea, these trying to get an understanding of what a consortium can do in terms of exchanges in principle. But then the next step was to go towards more explicit models for consortia, namely trying to consider the environment. So what is usually done is something like the model of a fat batch, batch or fat batch process. So you have essentially your biomass X, concentration C, and we assume that there are certain fluxes into the cell and out of the cell, and then we also get growth. Because this is all in steady state, This essentially only holds when we make a quasi-steady state assumption. This is now the same over time. We have decreasing concentrations of nutrients, increasing concentrations of biomass, and this holds in quasi-steady state over this exponential growth phase. So we were wondering to what extent this assumption on the environment actually has an influence on the outcome of the predictions. So we made the other possible alternative, which is also much more relevant for consortia like our microbes in the human gut, and that's the chemostat scenario. So we have a certain flow in, so D is the dilution rate, Cn is the input concentration of a particular substance we're interested in. Again, we have our microbes living in here, multiplying, but then we also have the outflow with the same dilution rate D that carries away both biomass as well as potential nutrients. So the point here is that really the environment should be explicit because the steady state concepts differ between these two assumptions here. So then we went and tried to look at what are the possible scenarios that essentially tell us what decisions a community could do. And there are classical models from game theory, such as the prisoner's dilemma. This is shown here on the left. So this is usually characterized by a payoff matrix. And here we have these two players, 1 and 2. They're in prison and they get the highest reward. So this is indicated by this 3T, the highest global reward if they both cooperate. So in this case, don't admit anything. If one defects, this one gets an even higher payoff. If both defect, then that's in the middle. So the nice thing is that this can be formulated also as a metabolic prisoner's dilemma game. So in this particular example here, we have two microbes. This is shown by the different backgrounds here. One is better at producing substance A. The other one is better at producing substance B. The optimal solution, the globally optimal solution, would be that they exchange the substance they're not good at producing and take this up from the other partner. But then the question is, do we have decision-making essentially at the level of the community, which would we then expect to lead to this optimum, or the individual or agent decisions, which in most cases would lead to this defective suboptimum. So we try to formalize this now in the metabolic context, and also in the view of these two different environments, batch, which is B, and C is for chemostat. I'll show you only the chemostat models here because they're more interesting. So the basic idea here is that we may have mass balances for this chemostat. So this is, for instance, for the biomass. So we have dilution and growth. If we assume a community objective, then it's formulated like this one here, similar to OPCOM. So just maximal community growth. And then we have the constraints on the individual microbes here, as well as constraints on the individual components, so metabolites in the medium. The agent model looks very similar, but here now we have one single objective, namely to maximize the growth of each species individually. So these are essentially prototypes for decision-making, just to see what the corner cases are. And the nice thing about this here is that we can formulate this such that it can be solved analytically. So essentially via KKT reformulation and then optimization leads to a mixed integer linear profile. So this is the outcome for this metabolic prisoner's dilemma. And what is interesting is whether the two species exchange A and B or not. So these are the highlighted rows here. Then we have the chemostat. CA means agent, so individual objectives. They don't cooperate. If we assume a community objective, then they cooperate. And if we assume community objective in batch, they also cooperate, but with a different solution. So here, the bottom lines, we get cross-feeding or cooperation only when we assume this explicitly. We can also ask different scenarios. So for instance, can we achieve coexistence? In this particular case, different formulation here. We have different substrates, A and B, provided externally, and one uses more B and the other one uses more A, but they don't need to communicate. They don't need to cross-read. Again, here the solutions, this is a bit more complicated here, depends on the ratio of A and B in the medium. This is on the x-axis here always. And then we have steady state solutions where we have only one of the two. So x1 or x2, these are the green lines, and we have coexistence solutions here in the middle. So depending on the environment, we either get single species or a coexistence of both. And this works both when we assume that each species has its individual objective or when we have community objectives. So in the end, we tried to see whether this makes sense for real-world applications, and we turned to synthetic microbial communities. These are very simple. So this has been published over 10 years ago. The idea there was we have mutants of E. coli. Each mutant has a different amino acid synthesis pathway deleted. So for instance, this would be lysine, gene deleted. Each individual strain can grow on rich medium. But when you then dilute them and put them on minimal medium, then the individual strains cannot grow simply because they lack the amino acid. But in some cases, you see synergistic growth, namely that in the scenario that one mutant produces one amino acid, the other one takes it up and produces the other lacking amino acid. So in order to make this feasible, we looked at core models, meaning just small models, 72 metabolites, 95 reactions, and then looked at pairwise consortia because there the data is there, so we could compare this to existing experimental data. So one result of this here is shown here. So this is the scenario where we assume we have growth in chemostat and a community objective. We saw for these steady states numerically, what you see on the left-hand side is the abundance, biomass of one of the mutants for alanine, the other one for glutamine. And the scale here shows you the residuals from the optimization. So the real solutions we're optimizing to achieve the steady state are somewhere between 10 to the minus 7. And this is on the accuracy of the numerical solver. So we get 4 solutions. One is the trivial one, 0, 0, no biomass. We get 2 solutions where we have single species. So on the one hand, this ala-oxytroph, on the other hand, this glutamine-oxytroph, and then one coexistence solution. And you also see that they have different external environments that's explicitly contained in this optimization problem. For instance, not only the biomass is different, we also have different concentrations of alanine or of glutamine. So that was essentially what you would expect. What would also come out of other approaches, for example, if you run dynamic FBA on this type of community model. What was really interesting, and we're still trying to figure out what happens exactly there. We now assume, again, we're in chemostat, but now we have individual objectives for the species. So it's this agent model. Every individual species tries to maximize its growth. That's the underlying assumption. We get again the trivial solution, so no biomass, 2 single species solutions, this one and this one here. But then the most surprising part of the solution was that we get a band of coexistence solutions. And where to land in this band essentially depends both on the details, how we model the dependencies or potential dependencies between fluxes and external concentrations, but also the initial conditions. And the interesting part here is that you essentially cannot find this solution bands by alternative methods, at least not in a reasonable way. So that's essentially what I wanted to show today. So the basic idea here is that even if you come to more complex microbiomes, this doesn't always require challenging computational methods. Like for this IBD case, it was essentially just reaction counting. But I think in general, the challenges are quite intricate. So one proposal is to use these minimal pathways to help explore and predict what exchanges can be possible in real-world settings, and the emphasis on real-world settings, because that approach essentially scales to, as I showed you, combinations of networks that have on your order of 30 to 50,000 reactions. And then we looked at these more explicit models, and they essentially highlighted a lot that it should be important to both characterize the environment and the decision-making assumptions explicitly, because otherwise this may highly influence the results. And there are many important challenges that I would be happy to discuss in a minute. And then the acknowledgements. So for the first part, IBD, this was Charlotte Ramon Boyes, who has since graduated. In the second part, this was Axel Theorel, still a postdoc in the group, and I'm happy about the microbiome collaborations, these people here on the right, and funding by the Swiss National Science Foundation. And that's the disclaimer.
Thank you very much, Jörg, for this very nice presentation.